Next, we're going to look at spermatogenesis, which begins about age 14 in males. Spermatogenesis is essentially meiosis in the male reproductive system. Sperm or spermatozoa, which are produced in the seminiferous tubule, are actually gametes. Another gamete would be ova or eggs from the female reproductive system. Gametes have 23 chromosomes. They have a haploid chromosomal number, which is half the amount of chromosomes of most body cells. Most body cells have 46 chromosomes, which is a diploid chromosomal number represented as 2n. The haploid chromosomal number is represented as n. Diploid chromosomal number has 23 pairs of chromosomes, one maternal and one paternal chromosome, making up a homologous chromosome pair. Next, we're going to look at meiosis. Meiosis is essentially gamete formation. In males, it's spermatogenesis. In females, it's oogenesis. And meiosis does differ from mitosis. There are 2 divisions in meiosis and only 1 in mitosis. We produce 4 daughter cells in meiosis via meiosis 1 and 2. Those 2 cell divisions that only include 1 round of DNA replication. In mitosis, we produce only 2 daughter cells. In mitosis, we produce clones of the original mother cell, whereas in meiosis, we produce 4 different genetically variable daughter cells. The functions of meiosis are to have the chromosome number from diploid to haploid and introduce genetic diversity. Some of the things that can introduce genetic diversity are the variability of gametes because the homologous pairs randomly align during prophase 1, the crossover that can occur when different homologous pairs synapse. And so no 2 gametes are going to be exactly alike, and they're all going to be different from the original mother cell. Here we have a comparison of mitosis and meiosis. We can see that for mitosis, we get only 2 daughter cells, and they're both diploid just like the parent. For meiosis, after 2 rounds of cell division, we get 4 daughter cells, and they're all haploid, which is half the genetic material that we found in the mother cell. So we half the DNA during meiosis. We do not do that in mitosis. So there are 1 division in mitosis, 2 in meiosis, no synapsis in mitosis, but that occurs during prophase 1 of meiosis, and we'll see that tetrads or homologous pairs line up and allow crossover. That introduces genetic variability. We get 2 daughter cells from mitosis, 4 from meiosis, so we'll produce haploid cells by the end of meiosis 2. The role of mitosis is to produce cells for growth and tissue repair as a multicellular adult develops, and this ensures genetic makeup of all the body is basically constant. And then the purpose of meiosis is to produce cells for reproduction, or gametes. So that introduces genetic variability in the gamete and reduces chromosomal number by half, so that when fertilization occurs, we have 2 haploid organisms coming together to form a diploid chromosomal number. So meiosis 1, the first cellular division in meiosis, is actually a reduction division. By the end of meiosis 1, we will already have haploid cells. We will have reduced the chromosome number from 2n to n. Different functions and processes that occur in meiosis 1 include synapsis, which is aligning up homologous chromosomes that form pairs. They contain 4 chromatids, which are called a tetrad. Crossing over also occurs during meiosis 1. This is the exchange of genetic material between male and female chromatids. They create unique chromatids and unique chromosomes. The crossover occurs at chiasmata. At the end of meiosis 1, each daughter cell will have 2 copies of 1 member of each homologous pair, either maternal or paternal, and none of the other. So we'll have haploid chromosomal numbers in the cells produced at the end of meiosis 1. At the end of meiosis 1, we only have 2 cells, which will then go through meiosis 2 to produce 2 cells themselves, giving us a total of 4 daughter cells. Meiosis 2 is called the equational division because we're not going to half the amount of DNA, it'll stay the same. So we'll start with sister chromatids from meiosis 1 separated so that we'll get 1 per cell at the end. Now, like mitosis, we're going to have this is really going to be like mitosis, except we're not going to have any DNA replication before meiosis 2 starts. Otherwise, it's exactly the same as mitosis. So here is meiosis. we see PMAT1, metaphase 1, anaphase 1, and telophase 1. That's our first round of division. This first round, we'll see the DNA go from 2N to N. So these two daughter cells that enter meiosis II are haploid, and they'll stay haploid all the way down to the end. The equational division, or meiosis II, looks a lot like mitosis, except there's no DNA replication at the beginning of it. So let's go back and look, or specifically apply this to the male reproductive system. So that process, meiosis, in males is called spermatogenesis, and it occurs in the testes, specifically the seminiferous tubules where sperm are produced. In the seminiferous tubules, we have a stratified tissue, or epithelium, that gives rise to sperm. Most of those cells were spermatogonia, or spermatogenic cells. So again, they're located in the testes, they're in the outermost tubule parts or cells of seminiferous tubules. And this is where they remain prior to puberty. Until puberty, mitosis is all that's going to occur at spermatogonia. Spermatogonia themselves are stem cells, and they don't start to produce cells that can become sperm until the start of puberty. So meiosis of spermatogenic cells does not begin until puberty. At puberty, spermatogenic cells, like a spermatocyte, undergoes meiosis I to become two secondary spermatocytes. Secondary spermatocytes are already haploid, but they can undergo meiosis II to form 4 haploid spermatids. So going from a spermatocyte to 2 secondary spermatocytes, that's the process meiosis I. So secondary spermatocytes are haploid, just like the 4 spermatids. There's also another process involved in spermatogenesis, which is spermiogenesis. This process is how spermatids, which are early sperm, become what we think resembles mature sperm. So it's a process that gets rid of excess cytoplasm and reorganizes things around the sperm to create what will eventually be motile sperm as it enters the epididymis. So here is a seminiferous tubule. So there's multiple in this image, but we have one centralized. We have many layers of tissue, so it's stratified epithelium. You can see the cells are a little more round near the outer part of the tubule. This is where the stem cell spermatogonia would be. And meiosis I would begin closest to the outer tubule, then proceed towards the lumen. So spermatids would be located closest to the lumen, and after spermiogenesis, the sperm would be pushed out of the seminiferous tubules to a straight tubule by those myoid cells contracting. So here we've taken just a piece of tubule. So here is our lumen. Here's the outer part of the tube. We'll call that the basal lamina. And before puberty, all that's occurring is these type A and B stem cells are just dividing. They're just performing mitosis to make more of them. At puberty, the type A daughter cell is going to remain at the basal lamina. The type B daughter cell is actually going to start to travel. It's going to start to travel towards the lumen. As it travels towards the lumen, it goes through meiosis I and II. The type B daughter cell, a stem cell, differentiates into a primary spermatocyte. A primary spermatocyte completes meiosis I and becomes or creates 2 secondary spermatocytes. It's the secondary spermatocytes that are already haploid, and then each one of them are going to go through meiosis II, which is like mitosis, so they each generate a spermatid. So we'll get 4 daughter cells total. The early spermatids develop into late spermatids, and through the process of spermiogenesis, these spermatids, which are round cells, start to look like spermatozoa. And the spermatozoa will then leave the tissue around the lumen and enter the lumen itself and then travel via the male reproductive duct system. So let's go through that again. So the mitosis of spermatogonia, what we're looking at here is the process of spermatogenesis, which begins at puberty. So spermatogonia are stem cells. They're going to be in contact or close to the epithelial basal lamina. And before puberty, we're just going to have mitotic division. So we'll get type A daughter cells and type B daughter cells performing mitosis, getting basically replicates of themselves. The type A cells maintain germ cell lines at the basal lamina, but at puberty, the type B cells begin to move toward the lumen and develop into primary spermatocytes. Primary spermatocytes are diploid. At the end of meiosis 1, the primary spermatocytes will have become secondary spermatocytes. We will have created two secondary spermatocytes, and secondary spermatocytes are haploid. So that was the reduction division. The secondary spermatocytes will enter meiosis 2, and each secondary spermatocyte will give rise to two spermatids. Now, we had two secondary spermatocytes, so if they each give rise to two spermatids, we'll have 4 daughter cells total. A spermatid is a small non-motile cell. It may not even have a flagellum yet, and it's going to be closest to the lumen of the tubule. So you can see here at the end of meiosis 2, we'll have our early spermatids. Then they'll go through a process called spermiogenesis, which occurs within spermatogenesis at the end stage of spermatogenesis. And that is going to turn our early spermatids into mature spermatozoa. So for spermatids, the correct chromosome number is always haploid or N, but they're non-motile. Through the process of spermiogenesis that occurs during late spermatogenesis, spermatids elongate, lose their excess cytoplasm, and form a flagellum, or a tail. That's when they become a true spermatozoon, or sperm cell. So here's the process of spermiogenesis. Here is an early spermatid. We see it starts to go through some changes. Here we're really starting to see the structure of sperm. We see these mitochondria start to line up along the neck region. So that neck region is where the mitochondria would be located. We're losing this excess cytoplasm. We saw early on the development of a flagellum or a tail. And the final spermatozoon that is created after approximately 24 days via the process of spermiogenesis will contain a head, midpiece, and tail. The head is the genetic region. It contains a nucleus but also a helmet-like acrosome that contains enzymes that are hydrolytic and are able to penetrate an ovum. The midpiece is where the mitochondria are. It's the metabolic region, so it has ATP that will be used to generate movement of the flagellum. The flagellum is also known as the tail. It's the locomotor region, and it proceeds to move back and forth or is whip-like and is powered by ATP. Now, we've talked about the spermatogonia and how they can become spermatocytes and then spermatids, but there's cells that the spermatogonia and the other cell types we talked about are retained within. And those are the large supporting cells called either sustenocytes or Sertoli cells. And they extend throughout the wall of the tubule. They surround the developing spermatogonia, spermatocytes, and spermatids. They provide nutrients and signals to dividing cells, and they move the cells along to the lumen. They also are the cells that secrete testicular fluid into the lumen for sperm transport once they leave the stratified tissue of the seminiferous tubule. They're also able to perform phagocytosis to get rid of any faulty stem cells or faulty germ cells, get rid of excess cytoplasm during the process of spermiogenesis, and they produce all those chemical mediators that regulated the process of spermatogenesis. The role of sustenocytes is highlighted by the fact that it divides the tubule into two compartments, the basal and adluminal compartment. The basal compartment contains stem cells, spermatogonia, and primary spermatocytes that are diploid. The adluminal compartment contains all meiotically active cells that are haploid, and they're going to be closest to the tubule lumen. So here we can see the basal compartment. I'll actually separate it out. So the basal compartment contains type A and B daughter cells, and it can contain primary spermatocytes. Not always. You can see the primary spermatocyte in this image is kind of right on that barrier. But the adluminal compartment proceeds from that basal compartment towards the lumen, and it contains all of the cells that are haploid and the cells that undergo spermiogenesis. The tight junctions between the abdominal and basal compartment form the blood-testis barrier, which prevents sperm antigens from escaping into the blood and thereby activating the immune system. But that's important because sperm are not formed until puberty, so they're absent during the immune system development. So if they were able to leave, basically if there were not tight junctions forming the blood-testis barrier, in males they would actually cause an immune reaction if they were able to leave the testes and the reproductive duct system. So spermatogenesis is meiosis in the male reproductive system. It takes about 64 to 72 days if conditions are hospitable, and it's the pressure of testicular fluid that is going to be pushing the immotile sperm into the epididymis. They will gain motility and fertilizing power in the epididymis. They do not have it when they enter. And a homeostatic imbalance we want to mention briefly is infertility. And we've seen a gradual decline in male fertility for the past 50 years. We think it's because of alien molecules called xenobiotics. Those include things like PVC, environmental toxins, phthalates, pesticides, herbicides, stuff like that, antibiotics, radiation, lead, and marijuana. We also think a lack of selenium or excessive alcohol abuse, lack of specific calcium channel, ions. Those things are all going to possibly play a role in infertility. There's, of course, anatomical obstructions, hormonal imbalances, and oxidative stress. Fevers, even hot tubs can play a role in the development of infertility. And that is it for Part A of the reproductive system. Make sure to go back and review video 1.